an extinct animal was born. In 2003, a cloned Pyrenean Ibex, or the Bucardo, took its first breaths after a C-section in Spain. Minutes later, it was gone. Lungs collapsed from defects no scientist could foresee. Receipts first, verdict later. Proof of concept or proof of limits? The claim was de-extinction made real. We brought back an extinct species. The actual lift was somatic cell nuclear transfer, SCNT, the same method used for Dolly the sheep. Researchers took frozen skin cells from the last female ibex, Celia, and fused their nuclei into enucleated domestic goat eggs. Once a blastocyst formed, it was implanted into a surrogate goat to gestate. Sounds straightforward, but every step was a lottery ticket. Cell fusion rate, embryo viability, and the surrogate's ability to carry a hybrid fetus. Out of hundreds of reconstructed eggs, only a few made it to term and only one to birth. So yes, the claim was technically true. A Bucardo was born, but the lift shows how fragile that truth was. A single neonate at the edge of biology and luck. Micro verdict: process works in theory, outcome fails in practice. The Bucardo clone remains the only extinct species ever brought to live birth. No viable line exists. The frozen cells used for Celia's clone have been degraded by time and freezing cycles, leaving scientists with limited genetic material to work with. Subsequent attempts to repeat the procedure stalled in the lab stage. Funding dry spells, ethical debates, and logistical barriers stopped the momentum. No second birth has been attempted since. In practical terms, the Bucardo experiment became a historical proof point, not a template. Modern programs like Revive and Restore now favour genome editing and proxy species approaches over full cloning. Micro verdict: legacy intact, continuation absent. Behind that single birth stood hundreds of surrogate goats subjected to surgery, implantation, and C-section delivery. Each procedure meant anesthesia, infection risk, and recovery time, a high cost for an experiment with near zero odds of success. The 2009 Theriogenology paper details 57 implants, producing 7 pregnancies and 1 birth. That math tells its own story. A dozen surgeries per trimester, hundreds of lab hours, and no offspring that lived beyond 7 minutes. Post-delivery, the newborn was placed in a neonatal ICU, intubated to assist breathing, but its lungs were malformed. Scientists watched the first heartbeat of a revived species fade in real time. Ethical balance comes into sharp focus here. How many living animals must endure risk for one non-viable extinct fetus? Micro verdict: scientific gains, yes. Ethical comfort, no. Even if the clone had survived, what then? The Pyrenean Ibex lived in the Aragonese Pyrenees, habitat now fragmented by roads and climate shift. Releasing a single animal into that would be symbolic, not restorative. The project's budget ran into hundreds of thousands of euros for facilities, equipment, and veterinary staff, with no foundation support after the initial headline. It ended as a case study in scientific limits rather than revival. Meanwhile, Spain diverted resources towards protecting the living Cantabrian Ibex, a closely related subspecies facing its own pressures. Micro verdict: budget spent, species not restored. The failure was not random. SCNT clones often suffer from epigenetic errors, genes failing to reset after nuclear transfer. That leads to placental malformations, and respiratory defects, exactly what killed the Bucardo clone. Placenta acts as an interface between species. When it's a hybrid goat ibex version, signals misfire. Attempts to fix this with chemical reprogramming or better donor mitochondria remains experimental. Until those glitches are solved, every clone is a biological coin toss. Genetic diversity poses another block. All known cilia samples represent a single genome. Even perfect SCNT would reproduce a species of one doomed by inbreeding from the moment it breathes. Microverdict: replication possible, resilience absent. Modern researchers see the Bucardo as a warning. Rather than resurrecting extinct icons, they're building genetic resilience into relatives that still exist. Think of it as editing for survival instead of revival. In Spain, Ibex genetic banks now focus on disease resistance and breeding diversity for the living species. Globally, projects like the passenger pigeon revival use gene edits in rock pigeons, less trauma for surrogates, more potential for self-sustaining flocks. The Pyrenean Ibex proved that de-extinction is not binary. Birth is one metric, survival another. Integration, the real goal. Until science can hit all three, every success story ends with a funeral. Microverdict: lesson learned. Mission incomplete. So, does the Pyrenean Ibex earn a green light for de-extinction? It delivered a world-first proof of principle, an extinct species born again by human hands. That's a scientific miracle worth acknowledging. But miracle and milestone aren't the same thing. When the result dies within minutes, it shows a boundary science has not yet crossed. Every attempt since has hit the same wall. Reprogramming errors, weak placentas, zero genetic pool. Until those barriers fall, Bucardo revival is symbolic, not sustainable. Verdict: veto. Birth achieved, survival not. Still pending. The Bucardo clone showed that even victory can sound like a flatline. But another species might break that pattern, the passenger pigeon, where the goal is a living flock, not a lonely miracle. Stay with the series. Next episode: De-Extinction, passenger pigeon, rebuilding the flock cell by cell. Thank you for joining me in this episode of the series.